So welcome back, whether you took a break or whether you just simply moved on to the next recording, welcome back again. Let's take a look at those concept check questions. So what molecules are the inputs and outputs of photosynthesis? If you use the simple equation, it's carbon dioxide and water that are the inputs and glucose and oxygen that are the outputs. If you use the more chemically and I guess biochemically correct equation, then you add water as a output as well, because it is not only a reactant, but it's also now considered to be a product of photosynthesis, to take into account what we know about the origin of the oxygen. Name the two stages of photosynthesis in the correct order. Well, seeing as how we need to capture light energy before we can do the reduction of carbon dioxide, it makes sense that the light reactions are the first stage and the Calvin cycle is the second stage, and that is the correct order for them. What molecules serve as the energy bridges between the two stages? Well, the transfer of energy from the light reactions is via the ATP as chemical energy, and NADPH is the source of electrons for reduction. What is returned to the light reactions is NADP plus and ADP and PI, so that they can be reconstituted as NADPH and ATP. So those are the energy bridge molecules. Hopefully you had all of those. So given that light energy is the source of energy for carrying out photosynthesis, we need to talk very briefly about what light actually is, because it's in understanding what light actually is that allows us to see how light energy can be used as a source of energy for driving photosynthesis. So the first thing to remember is that light is a small part of a larger spectrum of energy called electromagnetic radiation. And here you can see the full spectrum of electromagnetic radiation, with light more or less somewhere in the middle. And the different colors of light that we are able to detect simply reflect the wave nature of light, and more specifically, the wavelengths of the different colors of light that we can detect. Our eyes are sensitive to a set of wavelengths between around 380 nanometers and 750 nanometers. But it turns out that electromagnetic radiation has a wide variety of wavelengths, some of which we can detect in other ways. So infrared, which lies at a longer wavelength, so it's above 750s, you have red and then infrared. Infrared is heat energy. And so you can feel that on your skin. On the other end of the spectrum, at a wavelength shorter than 380 nanometers, is ultraviolet. And while we can't necessarily detect ultraviolet directly, we see its effect because that's what causes sunburn and skin cancer. If you're lucky, it might cause your skin to tan. But the problem with that is if your skin is tanning, you're getting skin damage anyway, because the tanning response is simply an indication that your skin is being damaged. That's the way it works. So there's no real healthy way of interacting with ultraviolet light. Whether you do it by lying out in the sun or whether you lie on a tanning bed, that ultraviolet radiation is going to damage your skin. That's what happens when your skin tans. Other wavelengths we can utilize, so radio waves, we still utilize for communication. If you listen to the radio, some of you may still, you're utilizing radio waves. If you are using your cell phone or using your microwave oven, or you're watching over-the-air television, not using cable, but watching over-the-air television, then you're receiving microwaves. Just as an aside, the reason why a microwave oven heats your food is because the wavelength of the microwaves that are being produced cause the water molecules in your food to vibrate. And of course, vibration is motion, and that's kinetic energy. And we know that temperature is a measure of the average kinetic energy. So if you cause the water molecules to vibrate faster or harder, you're increasing their kinetic energy, which means you're increasing the temperature. And the use of microwaves to heat food was actually discovered quite by accident. The original use of microwaves was in a radar system developed during World War II. And shortly after World War II, people were experimenting with making it more efficient. And apparently one of the researchers had a chocolate bar in his pocket. And when they turned the radar on, the chocolate bar melted. And someone figured out that there was a correlation between the radar and the melting of the chocolate bar. And the company would eventually go on and produce the first microwave oven, and it was called a radar range. It was about the size of a large refrigerator. Go figure. Anyway, we talked about infrared, we talked about ultraviolet. X-rays, very short wavelength, can pass through skin but can't pass through bone. And so we can use them to take pictures of your skeleton and hard tissue in your body. And we can also use them industrially for diagnostics of various things. Gamma rays, very, very short wavelength, very, very energetic, basically pass through everything except a solid block of lead that is about 4 or 5 inches thick. That will stop them eventually. But there are high energy gamma rays that come from out in the universe. We call them cosmic rays, and they basically blast through everything because they are so energetic. Now, if you've been paying attention, you'll notice that as things get more energetic, as we move from right to left, the energy is increasing, but you also notice the wavelength is decreasing. So there is a correlation, an inverse correlation between the wavelength and the energy. So when we look at visible light, the colors that our eyes are sensitive to, and which, by the way, the vast majority of life, including plants, are sensitive to, the wavelengths at the red end of the spectrum are the longest wavelengths and therefore represent the lowest energy level. At the other end, at the violet end of the spectrum, you have the shortest wavelengths and therefore the most energetic of the wavelengths. Now, most of you are familiar with light as a wave. When you look at yourself in the mirror, you have light bouncing off the mirror and coming to your eyes, and that's what you're seeing with. That's the reflection of a wave, and waves tend to reflect off things, tend to bounce off things. And under those circumstances, waves don't transfer energy particularly well because they tend to bounce off rather than transferring energy. So how then can light as a wave be used as a source of energy? Well, the answer is a little bit more complicated than you might think. But before we get to talking about just how that is, let's take a look at the wavelengths of light that are actually driving photosynthesis, because that's pretty old knowledge. It goes back to 1883, in fact, and a German biologist by the name of Theodor Engelmann. And Engelmann decided that he would use the current knowledge of photosynthesis and the current knowledge of light to try and figure out which wavelength was actually best at driving photosynthesis. This is one of the experimental asides in the textbook, so that's why it's at this position in the presentation, because this is where it occurs in the chapter. So how to go about doing this? Well, Engelmann knew that algae, and specifically Spirogyra, which is known as a filamentous algae, carry out photosynthesis and therefore must produce oxygen, because the production of oxygen as a byproduct of photosynthesis was figured out in the 1750s by an English scientist named Joseph Priestley. And Priestley figured out that a plant in light could revitalize air that had been devitalized by a burning candle or the presence of a mouse. He put burning candles and mice in glass containers and noticed that the candles went out and the mouse started to suffer. And if he left it there for too long, the mouse would die. But if he put a plant in the same container after the air had been devitalized, the plant, as long as it had sunlight, would revitalize the air. And he called this revitalizing factor oxygen. He didn't realize that he had discovered a new element. That would not be figured out for a little bit more time. It was after Priestley died, in fact. But Priestley had discovered the presence of oxygen, not only the presence of oxygen, but the fact that plants produce oxygen. So Engelmann knew all about this. He also knew of the work of Sir Isaac Newton back in the 1650s, using a glass prism, a triangular block of glass, to split sunlight into its component colors. The colors that we see here, I mean, these are the colors of the rainbow. The water droplets in the air do the same thing as Newton's triangular block of glass, the prism. It splits the light up into its component colors. So what we call visible light, or to use another term, oops, back, make sure the pen is active, what we sometimes refer to as white light, is actually made up of all of these component colors. So when we see all of these component colors together, we see the resulting light as white. But if you pass the light through water or through one of these triangular blocks of glass, a prism, the way the light interact, or different wavelengths of light interact with the water or the glass, causes them to separate out into separate streams, and we see the separate colors. And Engelmann knew about Newton's work with these triangular blocks of glass, these prisms. And so he knew he could split the light from the sun into its component colors. So here's what he set out to do. He put the algae, the Spirogyra, in a container and laid it out in strands. He then put the prism next to it and allowed the different colored light to interact with different parts of the algae. So now he had different colored light driving photosynthesis, or presumably driving photosynthesis in different parts of the algae. The question then became how to determine which parts of the algae were making the greatest amount of oxygen. Well, here he just simply turned to aerobic bacteria, knowing that they would cluster wherever there was a lot of oxygen. That would be their preference. So he introduced them in to the container and then put everything under a microscope and took a look to see where the aerobic bacteria were clustering. And what he discovered was that the bacteria accumulated specifically in the violet-blue end of the spectrum and in the yellow-red end of the spectrum, not very much in the green portion of the spectrum. And what Engelmann had discovered is what we refer to today as the action spectrum of photosynthesis. It correlates the wavelength of light with the rate of photosynthesis. And Engelmann's action spectrum is so good using these bacteria that we can't improve on it with all of our technology. We have machines called spectrophotometers that can generate action spectra. And even with all of that technology with the spectrophotometers, we can't improve on Engelmann's basic action spectrum. What the spectrophotometers give us is essentially identical to what Engelmann had discovered. And what he had discovered was that blue-purple light and yellow-red light drive photosynthesis most effectively. Green, not so much. So what that tells us is that green light probably is being rejected as a source of energy for photosynthesis. The question then becomes, why the green light energy being rejected, and why is it purple, blue, and yellow, red that's being accepted as a source of energy for photosynthesis? So this animation simply talks about the role of different colored light in driving photosynthesis. Basically, it says Engelmann was right. It is purple blue or violet blue and yellow red and not green. And then it talks a little bit about why. And so it has to do with a molecule called chlorophyll. And most of you have probably heard of chlorophyll. You may have been introduced to chlorophyll again if you've discussed photosynthesis at all in a previous class, perhaps in high school. So the chlorophylls are the molecules that we recognize as being the molecules that absorb the light energy. And it's because of the way the chlorophylls interact with light that leads to leaves being seen as green. Why? Because the chlorophylls will not accept green light energy. They reject the green light energy. They will capture the violet blue and they will capture the yellow red, but they won't capture the green. So the green is the only light that either gets reflected off the leaf or passes through the leaf because it's not captured by the chlorophyll molecules, which leaves only green light to come to our eyes, and therefore we see the leaves as being green because it's the only light that's available for our eyes to detect. The other colors have been captured by the chloroplast via the chlorophyll. And that's why leaves are green. In fact, that's why you see anything with the particular color that you have. So if we go back to this slide of the spectrum, why do we see red at this end? Because the computer screen is generating red light. But if we had an object that was red in the room, look around your room. Do you see something red? I can see something red. When I look at that, the reason why I see it as red is because the red is the light or the wavelength of light that's being reflected off that. But sunlight is falling on there. What happened to all the other colors, the purple through yellow and orange? Well, they all got absorbed by the pigments, in this case in the plastic, that's showing a red color. And so the only light that's available for me to see is the red light, the red wavelengths. We make the same argument with blue. We can make the same argument with yellow. Any color that you can see, you see because that's the colored light, the wavelength of light that your eyes are detecting. When you see white light, as I said, you're seeing all the colors together. And so you detect all the colors as combined together as white. So then what does black represent? Well, black represents all of the different wavelengths being absorbed and none of them being reflected to your eyes. So it is a, quote, black hole, a lack of light. None of the light is being reflected off that. All of the colors are being absorbed, and that's why you see it as being black. It's a lack of reflected light. So again, we're still talking about reflection of light. We're still talking about wave behavior. We really haven't addressed what it is about light that allows it to function as a source of energy, because we're still talking about waves, we're still talking about reflections, and we know that waves, when they bounce off things, when they reflect off things, don't transfer energy. So what is it about light, now that we've taken this diversion to Engelmann's experiment, what is it really about light that allows it to function as a source of light, of energy for photosynthesis? Well, it has to do with the fact that light has a dual character or a dual nature. It can function as waves. And when you're looking at light reflecting off something, like a mirror, you're seeing the wave behavior of light. But it turns out that light can also function as a particle. It has a particle nature. And those particles are called photons. And this is basically the same characteristics as electrons. Electrons can be drawn as particles, but electrons can also be drawn as clouds of negative charge, a wave behavior. And so what we're really talking about here is the particle behavior of light alongside the particle behavior of electrons. If a wave bounces off something, no energy is transferred. But when two particles collide, energy is transferred. And so when the particle of light, the photon, collides with the electron as a particle, there is a transfer of energy. And this is, of course, the first law of thermodynamics. As it is quoted here on the slide, energy cannot be destroyed, it can't be created either. And so some of the energy of the collision has to be converted to an absorbed form of energy in the electron. And this, in turn, turns out to be an increase in the potential energy of the electron. So how does that take place? I think I'm going, well, yeah, I can tie it together. I was just thinking about how I'm going to tie this together, given that we've got a slide here that talks about the chlorophylls. Well, how it ties together is because of the way in which the chlorophyll actually captures light energy. There is a portion of each chlorophyll molecule, and there are two different types of chlorophyll molecules called chlorophyll A and chlorophyll B, that are responsible for capturing light energy. It's referred to as a porphyrin ring, and there are no really good diagrams of a chlorophyll molecule in the textbook, unfortunately. But that ring has electrons circulating in the ring structure, and those electrons are very, very weakly held. And those are the electrons that are going to be energized by the light energy. Chlorophyll A is blue-green in color when it's isolated. And chlorophyll B is yellow-green. And as you might guess, because they appear to be different colors, they're absorbing slightly different wavelengths of light. And so when we take a look at how the chlorophylls absorb light, you can see that the two absorption curves of chlorophyll A and chlorophyll B don't exactly overlap. They do kind of overlap, but not exactly overlap. So chlorophyll A specializes in absorbing a lot of violet light and a little bit into the blue, but remember, it's blue-green in color, so it's not going to do particularly well in terms of absorbing blue. The chlorophyll B is yellow-green in color, so it's able to absorb more down into the blue, a little bit less in the violet, but some into the violet. But it absorbs mostly into the violet on into the blue. And when we get to the other end of the spectrum, it turns out that the chlorophyll A and B molecules also absorb energy down here. Chlorophyll A primarily in the red portion of the spectrum, and chlorophyll B more in the tail end of the red and a little bit into the orange, but remember it's yellow-green, so it's not going to touch much of the yellow. And of course, none of them are going to really absorb the green. There is a third type of pigment. It's a family of pigments called the carotenoids, and there are different types of carotenoids that turn up in leaves. These are bright yellow in color. These are the pigments that give carrots their characteristic orange color, not because the pigment is orange, but because there's so much of this yellow pigment that it appears to be orange in the carrot. So carrots are a really good source of carotenoids. Why do we think of them as being important? Because as you'll see, carotenoids, and particularly the type of carotenoid in a carrot, is a precursor to vitamin A. And so when we eat carrots, we get a source of a molecule that's converted to vitamin A. The carotenoids as yellow pigments also assist in absorbing light energy in the blue and the violet end. Why an emphasis on the blue and the violet end? Remember, those are the short wavelengths. This is the high energy end of the spectrum. So this is where we want to capture most of the energy, because this is maximizing the amount of energy that can be captured. It turns out, however, that because of the way the light is captured, the best way to measure the total amount of energy used by photosynthesis is to measure at the red end. So there's a whole lot of backstory to that, which I won't get into. But red light is generally considered to be the best measure or the best way to measure photosynthesis. And we do a lot of measurements at the red end of the spectrum. But you can see, if we were to draw a line that encompassed the absorption here and the absorption here, it would look very much like Engelmann's result, the action spectrum of photosynthesis. And when we use our technology and we use the spectrophotometer, that's basically what we get. Being able to correlate the areas of the spectrum that the carotenoids and the two chlorophyll molecules absorb, and being able to correlate that with the regions of maximum absorption in the action spectrum of photosynthesis, tells us that these are the pigments that are involved, because we can account for this big bump in the action spectrum by these absorption profiles for the chlorophylls and the carotenoids, and we can account for this big bump by the absorption profile of the two chlorophylls. And that's what tells us that these are the pigments that are involved. Still haven't really told you much about the absorption of energy, other than it's got to do with collisions between particles of light called photons, and particles of light, or particles of the electrons as particles. And that somehow it's got to do with changes in potential energy. Perhaps I should answer that question. I've teased you for long enough. And I've already mentioned the beta carotene. Lycopene is the red pigment that you are familiar with in some fruits like tomatoes and watermelons, peppers, anything that's red, strawberries, cherries and the like. They have lycopene. That may be a natural sunscreen. There's some real evidence that lycopene in the diet helps prevent sun damage. How much of that you can count on, I don't know. I do know that there's a little bit of evidence that it does help in preventing sun damage. I'll just say that. Don't think that you can eat a lot of tomatoes and the like, and then go and lay out in the sun and not expect to get sun damage and sunburn. That's not how it works. It does help prevent some of the accumulated sun damage, however, apparently. There's some evidence for that. And one of the other things, so seeing as how we're talking about pigments still, maybe we should talk about this one as well, because it's here. If you see the fall colors, everyone knows that fall colors feature yellows and oranges and reds. Well, you know what those pigments are. Those are going to be the carotenes, the lycopenes, and the likes. So why those colors? What's happened to the chlorophylls? Well, if you look at a chlorophyll molecule and you look at the porphyrin ring that I've mentioned previously, in the center of each porphyrin ring is a magnesium metal atom. And magnesium, it turns out, is a very difficult mineral to get out of the soil for plants. So given that most trees are going to shed their leaves to protect them for winter, because those leaves would get frozen and lost, to prevent damage to the trees, they're going to shed the leaves. But rather than waste all of the good magnesium that's found in the chlorophyll, the leaves first pull all the chlorophyll out of the leaves before they let them fall off. And of course, as soon as you take away the green chlorophylls, what you're left with are the carotenoids, and another group of pigments called xanthophylls, and the lycopenes and the like. And so you get the oranges and the reds and the yellows and all the varieties of colors associated with the non-chlorophyll pigments that are found in plants. So that's where the 4 colors come from. So finally, let's talk about the photon or the particle behavior of light and the particle behavior of the electron. So if we think in terms of electrons as particles, we've talked before about the fact that electrons exist in energy shells. So here are some energy shells. And down here is the nucleus of the atom. So let me put an electron in. I'll leave it in black at the moment. So if that electron is collided with or hit by a particle of red light, a photon of red light, there's going to be a transfer of energy. And that transfer of energy now allows the electron to move out here. And remember, each of these energy shells represents an amount of energy, potential energy. And so by allowing the electron to move from this energy shell to an energy cell further away from the nucleus, you've got an increase in the potential energy of the electron. If it's a photon of blue light, here's the photon of blue light coming in, and it collides with the electron. Well, remember, blue light is at the short wavelength end of the spectrum. That's the high energy end of the spectrum. And so you'll get it jumping out to here. Now, if you think about green light and where green light exists in the spectrum, it's in the middle. So if green light comes in, where the electron would jump to would be here, except there's no energy shell for the electron to go to. So because there's nowhere for the electron to go, if it accepts the green energy, nothing happens. The green energy is rejected. Now, once the electrons are out here, and that's the reason why green energy is rejected. Once the electrons are out here, we say that they're in their excited state. They're unstable. Remember, if you climb to the top of the playground slide, you're in an unstable position because you've got extra potential energy. You're high off the ground, so you can move from there, you can fall off, you can slide down the slide, So the electrons in their excited state are unstable. And if they are not captured in their excited state, they will come back to their resting state or their ground state, which means we lose the ability to capture high energy electrons. So to understand what happens next, now that we know that we can transfer energy to electrons by collision between the light particles, the photons, and the electrons as particles, and that allows for an increase in the potential energy of the electrons, we need to figure out how we can capture those electrons while they're still in their excited state before they shed their energy, this increased potential energy, and return back to their unexcited state or their resting state. And that is done in complexes of pigments. Now, we can also see this basic idea in isolated chlorophyll. So if we take chlorophyll out of a leaf, and it's easy to do that, you could use an alcoholic beverage at home to do that, because ethyl alcohol, the alcohol in alcoholic beverages, is one molecule that it will extract. And if you grind a leaf up in some cold vodka or something like that, you'll get a green solution. That's the chlorophyll that's coming out. If you shine light onto that isolated chlorophyll in exactly the right way, you'll see that the solution is not green, but red. And that red light is actually light energy that's being given off by the electrons as they return from their excited state to their ground state or their resting state. And that energy is being given off in the form of a photon of red light that your eyes can see. This process is called fluorescence. So that light energy that's being given off is correctly referred to as fluorescent light. And the book talks about another example, and you can see the example here, the chemical sticks or glow sticks. When you crack them open and allow the chemicals to combine together, what you're actually doing is creating a situation where electrons get excited. They go to their excited state, but they want to return to their resting state again, their ground state. And as they do, they shed energy, and that energy is released as photons of light that our eyes can detect. Another way that you can see this is a fluorescent light bulb, fluorescent light bulbs. So it's electrical energy in a fluorescent light bulb that's exciting the electrons in the gases that are inside the bulb. It's a fairly complex process, but ultimately light is given off in all the visible wavelengths, and that's why we see the light from the fluorescent bulb as being white. But it's the same basic idea. Excited electrons returning to their resting state, shedding the energy that they gained by becoming excited, and that energy is ultimately released as photons of different wavelengths of light that we can detect. And so we see the light, and we see it as white light, or in the case of the chemical sticks, the different colors, depending on what chemicals are mixed together to create the fluorescence. Now, of course, again, if we're thinking in terms of photosynthesis, we do not want the electrons to return to their ground state. We want the excited electrons. We want the energized electrons. So how are we going to capture this, capture these excited electrons? Well, that involves a complex of chlorophylls and proteins that make up something called a photosystem. And the photosystems are buried in the thylakoid membranes. Each photosystem, and there are two types of photosystems, as you'll see, basically are made up of the same basic concept. A surrounding cluster of proteins in which the vast majority of chlorophyll molecules are embedded, and they surround a central complex called the reaction center. In the heart of the reaction center are a pair of chlorophyll A molecules, and those chlorophyll A molecules, known as the reaction center chlorophylls, are responsible for generating the high energy electrons. Each chlorophyll molecule, remember there are two in the reaction center, each chlorophyll molecule generates one high energy electron when it's stimulated. Now, before those high energy electrons can return to their resting state, they are captured by a molecule referred to as the primary electron acceptor. It's extremely electronegative compared to the chlorophyll molecules. So as you might imagine, if you recall what electronegativity means, being electronegative, it has a strong attraction for electrons. Here are electrons that are available to be captured. So it does that. It captures the high energy electrons from the chlorophyll molecules. Those high energy electrons are then passed on into an electron transport chain, as you'll see. So this is basically how the energy of light is captured as high energy electrons. The photons of light energy collide with electrons in the chlorophyll molecules. There's a transfer of energy. Those electrons now are increased in potential energy, and they enter their excited state and move to an energy shell further away from the nucleus. The primary electron acceptor then captures them before they can return to their ground state or their resting state, their unexcited state. And so we have now captured the high energy electrons. But what's interesting as far as a photosystem is concerned is that only the reaction center chlorophylls can make these high energy electrons, but there are some 200 to 300 chlorophyll molecules. What do these remainder do? Well, they're acting as an antenna. So if you want an analogy so that you understand how this is working, think about a satellite dish. The dish is the surface, has a large surface area, and that's what's capturing the energy. But all of that energy is being reflected to the little box on the end of the stalk in the center of the satellite dish, because that's where the receiver amplifier is actually located. If the only energy that you captured was the energy captured by that little box, you wouldn't get much. But by surrounding it with a much larger dish that reflects the energy to that box, you increase the amount of total energy that's captured by the satellite dish. And that's basically what these extra chlorophyll molecules are doing. There's both chlorophyll A and chlorophyll B. They capture light energy themselves, but instead of creating high energy electrons, they transfer the energy from chlorophyll molecule to chlorophyll molecule until it enters the reaction center chlorophyll molecules, where it is used to generate high energy electrons. And this is some of the most efficient energy transfers that we know of. It's about 90% efficient in terms of the amount of captured light energy that eventually winds up in the reaction center. About 90% of the captured energy gets transferred to the reaction center. So it's incredibly efficient in terms of capturing light energy. So this is how a photosystem works. And as you'll see, it requires the collaboration of two separate photosystems to energize the electrons that originate in water in such a way that we have enough energy not only to generate ATP, but to also carry out the reduction of NADP plus to NADPH. So how does that actually take place? Because that's the last piece of the puzzle. Well, the photosystems are actually located within the thylakoid membranes. And if you want to read up those slides that I jumped over, by all means you can. You know, you can download the slides separately so that you can go over those. But rather than just rehash what's written on those slides, I want to talk about this slide, because it shows how the two photosystems act together to energize the electrons, to generate the ATP and create high energy electrons in the NADPH. So you'll notice here that it says first photosystem, second photosystem. That's ignoring the fact that there are two different photosystems. These are not identical photosystems. They are very different. The reason why they've done this is because the names of the photosystems It can get a bit confusing if you're thinking about the order in which everything acts. So this is called photosystem 1 or PS1 for short. And the reason why it's called photosystem 1 is because it was discovered first. Not because it's the first one in the chain, but because it was the one that was first discovered. This one is called photosystem 2 or PS2. And this was discovered second. And just to reinforce the fact that they are in fact very different, this has a maximum absorption at 700 nanometers. So it's in the red end of the spectrum. This has a max absorption at 680 nanometers. So they're very close in terms of the wavelengths of light that they absorb best, but they're not exactly the same. That's because they are different photosystems. So it requires these two acting together in concert to generate the energy or to absorb the light energy necessary to both make ATP and do the reduction of NADP plus to NADPH. So how do we talk about this? Well, this is called non-cyclic electron flow, because the electrons flow in a linear fashion from water through the two photosystems, eventually to NADPH. There is also a cyclic electron flow, but that's not really talked about in the book, and I'm not going to talk about that one because it's not really important for you to know that. This is the one that we really need to focus on. So the way I like to talk about this is to start with photosystem 1, because that's the source of the electrons that are used directly to reduce NADP plus to NADPH. So when this is activated by light energy, so light comes in, strikes the photosystem, it generates a pair of electrons. The electrons are captured by the primary electron acceptor and passed into an electron transport chain. So this is a short electron transport chain. And you should recognize those initials, ETC, for electron transport chain. And it passes the electrons on to an enzyme called NADP reductase, which tells you exactly what it's going to do. It's going to reduce NADP plus to NADPH using these electrons. This has lost electrons. Well, actually, I'm not going, let me not write that in because it might get confusing. So this photosystem has now lost electrons. And if we recall from cell respiration, LEOG, loss of electrons, oxidation. So these chlorophylls have been oxidized. And the unfortunate thing about oxidized chlorophyll is it cannot generate more high energy electrons. So we need to reduce it back to its reduced form, or the reaction center chlorophylls back to their reduced form in order to be able to continually generate high energy electrons. So those electrons come from photosystem 2. When it gets energized by light, it produces a pair of high energy electrons. Those are captured by its primary electron acceptor and passed off into an electron transport chain that connects the two photosystems together. Within that electron transport chain, you see the movement, I'm going to change color here, of hydrogen ions not from the matrix to the intermembrane space, but from the stroma to the thylakoid space. Probably not going to work really well there. Hopefully you can see that. And so this electron transport chain generates a hydrogen ion gradient across the thylakoid membrane, which is going to be used to drive the ATP synthase to make ATP. So that's how this flow of electrons, exactly the same as the flow of electrons in cell respiration, can be used to provide energy to make ATP. There's really no difference in the way it's done in chloroplasts versus mitochondria. In fact, experimentally, the role of the electron transport chain in the flow of hydrogen ions was actually demonstrated first in chloroplasts, not in mitochondria. I won't go into that, but that's where it was first figured out. That's where it was first demonstrated experimentally in chloroplasts rather than mitochondria. So now we have the electrons passing through the electron transport chain. Eventually they arrive at the photosystem 1 reaction center and are used to reduce the reaction center chlorophylls back to their reduced form again. But that, of course, has left the chlorophylls over here in the photosystem 2 reaction center in their oxidized form. We need to reduce those so that they can continue to supply electrons to photosystem 1. And that's where the water comes in. There is a complex of proteins called the water. Change that to red. There is a complex of proteins associated with photosystem 2 called the water splitting complex. And its job is to do what its name says, split water. It splits water into hydrogen ions, oxygen atoms, and electrons. The electrons go on to reduce the chlorophylls in photosystem 2 back to their reduced form. The oxygen atoms join together as to form oxygen gas and get released from the leaf. The hydrogen ions, well, they contribute to the production of ATP because the water splitting takes place inside the thylakoid space. So those hydrogen ions that are released from splitting water, they're sitting in the thylakoid space and they contribute to the generation of ATP. So they help in producing ATP. As we've seen previously when we've talked about processes, organisms do not waste anything. And here they're not wasting a potential source of energy in the form of those hydrogen ions. So they actually do utilize those as part of the generation of ATP. So you can see how all of this ties together to allow us to not only utilize light energy to generate ATP, something that some bacteria actually still do to this day. They actually use a photosystem and an electron transport chain to generate ATP using light energy. And it gives us a clue as to where photosynthesis may have actually come from in an evolutionary sense. Because if you think about it, if all you need to do to make ATP is lie out in the sun, that's got to be a real benefit, because you don't have to go chasing down organic molecules to make ATP. Just hang around in the sunshine and make as much ATP as you want. Ultimately, that would evolve into photosynthesis as we know it today, with two photosystems and the production of oxygen as we split water as a consequence of all of that. And there is some evidence to suggest that that is in fact the way it came about. Again, we don't really need to get into details, but it does give us some clues as to how photosynthesis, as the process we understand it to be today, may have developed over time through natural selection and evolution. It starts out as a process for making ATP using a single photosystem, but with the appearance of a second photosystem, we now have the potential to capture more light energy and turn it into something that is even more potent as a way of capturing light energy and storing it as chemical energy. So by way of summary of everything that we've talked about, here you can see what's going on. We have our two photosystems. So we've got PS1 and PS2. Might as well label them. Our electron transport chain that's transporting the hydrogen ions as the electrons pass through the chain. So remember, it's a pair of electrons that are passing through here, and as they move through, just as we saw in the mitochondria, we use hydrogen ions to balance that charge, and the hydrogen ions wind up in the space inside the thylakoid. And remember again, actually, let me do this way. Because this is something that the book has left out of this diagram. Remember, the water splitting also gives rise to hydrogen ions. So those hydrogen ions join the hydrogen ions that have come across as a result of the electron transport chain activity and contribute to the making of ATP. And you can see the ATP synthase also sitting there in the thylakoid membrane, utilizing the flow of hydrogen ions to make ATP. So now we have our energy sources. We've got reducing power in the form of electrons in NADPH, and we've got chemical energy in the form of ATP. Now we're ready to talk about the second stage, the Calvin cycle reactions that are actually going to utilize this energy to reduce carbon dioxide to sugar. But we will talk about that on the other side of the break. So we're going to take another short break here, or you may take a longer break if you wish, but I am going to stop the recording. And when we come back from the recording to the third recording and the last recording for this chapter, we'll look at these concept check questions before we move on. So what is the function of the reaction center of the photosystem during the light reactions of photosynthesis? What role do most of the chlorophyll molecules in a photosystem play? And what is the role of water as a reactant in photosynthesis? I'll see you on the other side of the break with the answers to those questions.